So we will continue from where we stopped the last time. And what we discussed yesterday was some properties of helicases, and we understood that helicases can work both on DNA as well as RNA, and they are required to unzip double-stranded DNA or double-stranded RNA, or even DNA RNA duplexes. And for this activity, they use the energy of hydrolysis of the terminal phosphates from NTPs. So it could be ATP, it could even be TTP, as we have seen yesterday. The, the hexameric helicase from T7 was using TTP for the hydrolysis of TTP to drive the unwinding of DNA. And we also realized that helicases can work both by binding onto single-stranded DNA at the junction fork, for example, if we look at this. So they could be binding to the single-stranded DNA, one of the single-stranded DNA, and unwinding it, or they could be binding at the junction fork on this double-stranded DNA, causing causing the separation of the two strands. We also saw that the directionality of the helicases could be either 5' to 3' or 3' to 5', and that the helicases can act as monomers, can act as homodimers, heterodimers, as well as functional oligomers. And there are a class of helicases which belong to the family of AAA ATPase. So they form a hexameric structure. And yesterday we had two questions. One was from Ratnesh, which was something like, if there are so many helicases in the human genome, does it mean that all the chromosomes encode for it? I'm sorry, Ratnesh, I completely forgot to look through this answer. But I will search again and I will get back to this. For Sarika, she had a question which was regarding whether all 6 subunits, so we showed a cartoon, and I'm going to play that cartoon once more. So here is the cartoon, which sort of depicts how the AAA ATPase hexameric helicases behave, and they took a specific example of T7 helicase. But this is a general cartoonic depiction because T7 is dependent on TTP hydrolysis, but here they have used an analogy of ATP. And what they suggest is that at a time, two of these monomeric units in this hexameric helicase would be either in an empty conformation, that is, they do not have a nucleotide bound to it, or they could be bound to NTP, or they could be bound to NDP plus PI. And Sarika's question was, is it always the case that you should have 2 of these bound? So I did look through it and I found a PNAS paper where they have solved structures, and they found that 1 or 2 subunits at a time could be bound by NTP. So that's as far as I could see in the literature. I'm going to look through it further. So Sarika, if you also come across something, you can contribute. Ratnesh, I will come to your answer in next week. Okay, so with this brief introduction, let's move a little bit more into helicases. And so one of the So let's take an example of a helicase, because there are just too many helicases and we cannot go through individual helicases. So I'm just going to pick up another helicase and try to look at how it functions and how we could study its functions and what tools could be used for this. Of course, single molecule tools such as optical trapping or even magnetic tweezers are very useful to understand how helicases function. And as I have mentioned, we will have a special class on magnetic tweezers the coming Wednesday by Dr. Saurav Raj. So he will explain it in greater details how magnetic tweezers function, and then you can understand how it is useful to study motors which have a rotary property, which rotates. All right. But for the moment, apart from T7 helicase, where we were looking at a helicase which has a hexameric structure, now we shift to a helicase which can act as a monomer and possibly also as a dimer, but it's a monomeric helicase. So the helicase that I've chosen today is called this PCRA, and it's a bacterial helicase, and it is an ATP dependent DNA helicase. So monomeric helicases have certain advantages to study them, because you can just make one mutation and that's enough and bacterial helicase on top of that, because in bacteria you have a single copy of each gene, right? You are not looking at duplicate chromosomes. So you're looking at one single chromosome, you have one single gene, and if you mutate that gene, you can study these molecules even further. And if it is a single molecule of helicase, then it's much easier to do manipulations at the level, at in vitro level, because you just have to assemble, you do not need to assemble an entire complex. It's only about getting that one protein. So it makes study of helicase properties a little easy. So this is one protein that has been quite widely studied. It's called PRCA. As I said, ATP dependent helicase, monomeric, present in bacteria. And what it does is it moves from the 3' to 5' direction on single-stranded DNA, and it has a translocase activity. What's translocase? It can translocate DNA. So as we saw in the last class, you have helicases that can sort of pull DNA, single-strand DNA strands through them. So such kind of activity is present in PCRA. It can loop out single-stranded DNA through it. So that's a translocase activity which is associated with PCRA. It's of course a helicase, so it can unwind duplex DNA, and the directionality is 3' to 5'. So with this little bit of a background, let us look at this at an earlier study when single molecular studies were not so popular. At that time, PCRA was being studied, and this group tried to understand the kinetics of how this PCRA functions. So if it is a helicase, it is not just unwinding DNA, but it is using an energy source. So here the energy source is ATP, and so every time it has to function, it has to utilize the energy source. So it has to break down ATP into ADP plus inorganic phosphate. And the inorganic phosphate must be released from the pocket. An empty pocket must be created for the next ATP to bind, which means that every cycle that it works, it is going to be utilizing ATP. So just by directly looking at the number of molecules of ATP utilized, how can you do that? You can do that by quantitating the number of molecules of inorganic phosphate that is produced. If you can somehow measure the number of molecules of inorganic phosphate that is produced, you can look at the rate of progress of this reaction. Rate of progress of PCRA dependent PCRA translocation on single-stranded DNA. So the experimental model that they took was this. They had a deoxythymine stretch. So D stretch, and 16 such nucleotides of thymidine were added one after the other to give you one single-stranded DNA stretch. And it was known that this helicase can bind and translocate on single-stranded DNA. So helicases have this two distinct properties, the property of unwinding DNA as well as translocating on DNA. Now these two properties are combined when we think of say an RNA helicase or a DNA helicase, because it has to unwind, but it cannot be static, because it would then cause a local unwinding, and then that would be stuck in that place. So it needs to also translocate that DNA or translocate the RNA so that the unwinding can progress through the chromosome or through the RNA structure. So here they were studying specifically the translocase activity. And as I mentioned, the translocase activity is studied by looking at the rate at which inorganic phosphate is released. Now, if you would like to know how did they measure the inorganic phosphate, so as I mentioned, this is an old paper, and so they were using quite different techniques to measure these. So they were using a chemical reaction which couples the release of released inorganic phosphate to produce another fluorescent moiety. And so they are actually measuring the amount of fluorescence that is emitted, because the amount of inorganic phosphate that's released is being used up to produce this fluorescent molecule. So in this manner, they were able to track how much of inorganic phosphate is released. So they have a system where they have taken this single-stranded thymidine stretch, single-stranded DNA, and they have taken this purified PCRA protein, they have added ATP, and now they are looking at the increase in fluorescence with time. Increase in fluorescence would indicate increase in inorganic phosphate release, which would indicate that the ATP is being utilized and therefore your PCRA is translocating on the DNA. So if you look at the curves here on A, you would see that there is a lag phase. That is, initially, there doesn't seem to be much that's happening. There is a lag phase. And then there is a rapid increase. And then there is some kind of a saturation, that increase, the rate of increase is not sustained, but rather it sort of plateaus over time. It sort of kind of saturates up over time. So how can you explain this entire phenomena? So you can explain this entire phenomena by thinking of it in this way. So what they had is a stretch of how many, 12 or 16? Okay, 16 nucleotides of thymine, single-stranded. And they have added to this a mixture of PCRA and ATP. So as soon as they add all of this together, there would be a time when time required for the PCRA to start binding on these single-stranded DNA structures, and then it would start translocating. So that time required for the PCRA to bind to single-stranded DNA and start to translocate is indicated here in this lag phase. So if that is true, if you premix PCRA and this single-stranded DNA, you premix it so that the loading of the PCRA on single-stranded DNA has already occurred, but you do not add ATP. If you do not add ATP, it will just load, but it will not translocate, right? It's not going to translocate, it's just loaded. So if you add a preloaded complex, and then add ATP, then there should not be a lag phase and it should move instantly. So that's what they see here. They see an instant movement where there is no lag phase if they use a premixed complex. And so what do they expect? They expect that every time the PCRA translocates on DNA, it's going to utilize ATP, and therefore you're going to have a release of inorganic phosphate, and this should be a rapid phase. So the initial loading is a slow phase, but there would be a rapid increase in inorganic phosphate. However, once this molecule, this PCRA, reaches the end of DNA, it has translocated throughout. It's a 3' to 5' direction translocation. It's translocated throughout the DNA, and now it's reached the end, to the 5' end. And it does not have any more DNA to translocate. So if it is not translocating, there are two scenarios that could happen. One is that it can just fall off. It can fall off from the 5' end. Or, I think you saw this in case of dyneins or kinesins, don't remember exactly, where we were looking at biological motors and futuristic design. You had seen asters forming, and I think kinesins, asters were forming, and the asters were forming because at the end of the microtubules, the kinesins would all accumulate towards the plus end and would stay there and not fall off. So if you think of a similar analogy, one is that the helicase can reach the 5' end and fall off. The other is the helicase has reached the 5' end and has not, and doesn't fall off. but it stays. If it stays, it may still be able to hydrolyze ATP. It has the ATPase activity. It may still be able to hydrolyze ATP without actually translocating DNA. If that is the case, now translocation of DNA might increase the rate of ATP hydrolysis, whereas if there is futile cycles of ATP hydrolysis, then this may not be at the same rate at which translocation dependent ATP hydrolysis was taking place. So these were the hypotheses when they looked at this data. So here, these are the hypotheses that they're putting forward to explain this data. So the lag phase is well explained. If you add a premix, there is no lag phase. You see a burst of inorganic phosphate release as the molecule is, as the helicase is moving through the single-stranded DNA. As it reaches the end, it sort of saturates out. Now, if you add more amount of this, if you add more of the DNA, then you would see a higher levels of this inorganic phosphate release. And inorganic phosphate release, but it would, if it is true that it can stay at the end and it can cause this futile cycles of ATP hydrolysis, then even if you add very high amounts of DNA, at some point this futile cycle will be reached, but will be reached much later. So those plateau regions will be reached later, and that is what you see here. As you keep increasing the amount of DNA. Whereas at low concentrations, you can see the plateau might have reached earlier. So these were really initial experiments that were trying to determine what is the nature, kinetics of these helicases as they are translocating on DNA. Okay, here is another interesting observation. So from similar kind of experiments, now they have moved a little further. So what they did here is that they took DT16. What if you change the 16 to different sizes of single-stranded DNA? You can make it 18, 16, 14, 12, different sizes of these single-stranded oligonucleotide stretches. And then you do the same kind of experiment and you try to see what is the rate of ATP hydrolysis, that is a release of inorganic phosphate, and you plot curves. And what you see is that as you keep increasing the number of nucleotides on the single-stranded DNA, the inorganic phosphate released is progressively increased, which means that there is a direct positive correlation between the nucleotide length and the ATP hydrolysis, which again goes to show that these helicases are dependent on ATP hydrolysis, and perhaps at each step of its movement, it requires ATP hydrolysis for it to move from one nucleotide to the next, to the next, to the next. Okay, and then they could also quantitate the duration of the rapid phase. Which one is the rapid phase? This phase. So how long is this rapid phase? The rapid phase would be longer if you have a longer stretch of nucleotide. So apart from the first few anomalous data, which is for like 4 or 6 nucleotides, where they could not get this kind of a linear relationship, maybe because 2-4 nucleotides is just too short, maybe the helicases attach and detach very quickly, or maybe they proceed. There are some errors if you consider very short nucleotides, but if you remove the oligonucleotides, if you remove that, throughout the rest, you see again a linear relationship. And from this, they kind of tried to find out how many ATPs are required for this helicase to move from nucleotide position 1 to nucleotide position 2. And in their calculation, they have found it to be half. But in reality, it is actually 1. So this is a rough estimation. You can understand these calculations are mostly a rough estimation, but they have more or less come to it. For them, it turns out to be half, but in reality, it is 1. So 1 ATP is required for this molecule to complete one cycle. So far, are there any questions? Any questions? Because I will continue with the same molecule but in a different, from a different paper now. No? Is it clear or is it not clear?
Clear, ma'am.
Wait, let
Clear,
me
ma'am.
increase the volume. Yes, Sarika, go ahead.
Yes, ma'am. It's clear.
Okay, thank you. So, okay, these were like very, I shouldn't say primitive, but kind of primitive because they were using very age-old techniques to try and get an estimate of the kinetics of the reaction. But with the advent of single molecular biophysics, it became much easy to understand how these helicases function. And as I have mentioned, we are not going to look at optical trapping or magnetic tracers now because we'll have a separate class on that and we'll have an expert to talk on that. Instead, what we are going to do is we are going to look at how the same molecule, PCRA, was studied using FRET. Now, before we go into that, let me ask you, what do you understand by FRET? Foster resonance energy transfer or fluorescence resonance energy transfer? Have you heard of it? And what do you understand by that?
Ma'am, it is something like something called like thread paint. It is used for like uh,
It's
what is
something
called
like,
fluorescently labeled labeling this DNA different regions of specific regions of DNA and visualizing that inside the cell.
no, what is this? It's a there's a physics behind this. If you can explain that, where it is utilized, you can utilize it in hundreds of things. But what is the physics behind it, Ratnesh? How much do you know about that? Sketchy idea?
I do not
Anything? No?
know, ma'am.
Ma'am.
Yes, Sarika.
Ma'am, it is a phenomena in which when we have two molecules which are in close proximity, say 10 to 100 nanometer, so if the emission intensity of one molecule is acceptor and another is a donor, so if the emission intensity of acceptor coincides or overlap with the absorption intensity of donor, so there is a phenomena observed that is called fret, where is the energy transferred happen.
और थोड़ा explain कर सकता है कोई? Anybody else with another try?
Ma'am, I would like to just add one thing. Wavelength here it is if the two molecules are in close proximity, then the wavelength absorbed by the one which is acceptor, and again it will emit when it will come to the ground state, it will emit certain energy or certain radiation of specific wavelength which will coincide with the wavelength absorption of the second molecule which is sorry acceptor, but first one was the donor. Sorry, I was confused. So when it will coincide with the absorption spectrum of the acceptor, so it will accept the that wavelength of energy that is emitted by the donor molecule, and after accepting that it will again come to the ground state and will emit certain amount of energy. Then the eventually the color or the something fluorescent or signal that is given will be equal to the emission wavelength of the second molecule and not the first molecule. And if they both are not in close proximity, then the emission spectrum will be equal to the first molecule emission wavelength. So it will show that they are not interacting anymore, and if they are interacting, then the emission wavelength will be of second molecule.
Okay, let me ask this question. Between Kavan, Sarika, and Farman, how many of you have taken a course in biophysics this time, or after joining
No,
IIT?
no, ma'am.
I haven't, ma'am.
Sarika, have you taken? in biophysics?
Yes, ma'am, I have taken.
Okay, थोड़ा बेटा ध्यान से पढ़ो because ये आपके PhD के लिए directly important है. Ratnesh, so do you have a course in biophysics?
Yes, ma'am. There is elective course, but I have not done it yet.
Okay, so if you have not done it's good to do it maybe if you have an elective course in biophysics because it definitely see uh, the amount of ideas that I can give on fluorescence or on FRET is also limited because I'm not a physics background person. But biophysics courses are taught by people who are physicists. And therefore it's good to understand this from somebody who for whom it is the niche area. My niche area is motor proteins. I am teaching motor proteins. But I can also give you an idea about FRET, but I can't give you the details of FRET. So all of you, I suggest please take a course in biophysics. It is going to help you all. So let me first go here before I go into anything else. So what you see here are two spectra. Two spectra. ये एक दो molecule है, Psi3, Psi5. That's just the name. Forget it for now. You have two molecules which have a characteristic excitation spectra and a characteristic emission spectra. So what is an excitation spectra? What is an emission spectra?
Ma'am, the light it absorbs should be considered as excitation spectra.
Okay, so the range of wavelength of light that can be absorbed by this molecule that depends on the electron configuration of the molecule. The range of wavelengths that this molecule can absorb would give you the excitation spectra. Now, the configuration, the electronic configuration would tell you at which point the absorption would be maximum. Okay, so for molecules you can have very sharp excitation spectra, very broad excitation spectra. It will depend on the electronic configuration of the molecule. So if you tweak a little the molecular structure, you can change the excitation spectra. All right, what is an emission spectra?
Emission spectra will be the when these electrons go into like a resting state or like to the state in the orbit which is which have lower energy, so they emit this light or any other wavelength, electromagnetic wavelength. So that that spectrum will be considered as emission spectra.
Okay, so the light that they absorb, that would push these electrons to say a higher energy state, but the higher energy state is a very short-lived state, and it immediately comes back to the ground state. But when it comes back to the ground state, because it doesn't come to the very same state, so there are some vibrational relaxations which ensure that even though it comes back and it finally gets into the same state, but the energy difference that is emitted is never the same energy that was absorbed. So the photons that it absorbs, the energy of those photons is higher compared to the photons that it releases, emits. So that's fluorescence. So this, the wavelength that it can emit can also be can also show a broad range or a narrow range. Again, it depends on the electronic configuration. Some molecules can have a very narrow range of emission spectra. Some can have very broad range of emission spectra. For example, many of you may have heard of the molecule DAPI. DAPI is used to stain DNA and stain nucleus. So if you want to see nucleus very clearly under a fluorescence microscope, you use DAPI. DAPI has a very broad emission spectra, very broad emission spectra, whereas other molecules can have very narrow emission spectra. It all depends on the electronic configuration. So okay, now one thing that was mentioned by Sarika and Farman was that you need 2 fluorophores. 8 fluorophores से foster resonance energy transfer नहीं होगा. So you need at least 2 fluorophores. Now, this energy transfer between these 2 fluorophores would depend on a number of aspects. Primarily, there are 3 aspects. One is that the excitation emission spectra of the 2 fluorophores should be nearly matched. How nearly matched? So here this is the excitation spectra of molecule 1 and its emission spectra. And this is the excitation spectra of molecule 2 and its emission spectra. You see that the emission spectra of molecule 1 here, it overlaps with the excitation spectra of molecule 2, and the overlap is shown here. So higher the overlap, greater is the efficiency with which this, if you excite this molecule, you may be able to excite the molecule 2 simply because of the emission from molecule 1. So this emission here may be able to excite this molecule 2. So that's the first thing. You need to have, an overlap of the emission spectra of one molecule and the excitation spectra of the other molecule. The greater is this overlap, the greater could be your energy transfer. But that is not all. You see, there is, in the name itself, you have this foster resonance energy transfer, and you may have heard the term foster distance. Has anybody heard the term foster distance?
Yes, ma'am.
Yeah. क्या होता है?
It's the minimum distance that is required for this process of Fred to be happen.
Right. So, which means that if you have the two molecules very far apart, you cannot have this this energy transfer. So it's a kind of energy transfer because you're looking at energy that is emitted by molecule 1 when it is excited. It emits energy, and that energy is utilized by molecule 2 to be excited. You're not separately exciting molecule 2. So then these two molecules should be very close to one another. And if you look at the equations of this of Fred, you would see that the energy the energy that is transferred from this molecule 1 to 2 is inversely proportional to the distance to the power 6, which means that at very close proximity you would have very high energy transfers, but as the distance moves apart, the energy transfer falls drastically, inversely proportional to r to the power 6, which indicates that you can very clearly understand if two proteins are interacting. A protein size would be somewhere roughly around how many nanometers?
I'm 10 to 100 nanometers.
100 मत बोलो, 100 too much हो जाता है. 10 nanometers, 7 nanometers, 8 nanometers, ऐसे ठीक. 100 nanometers, you're talking about complexes.
If we take it in angstrom, then it's 10 to 100 angstrom. Otherwise, that means it is 1 to 10 nanometer.
हाँ, okay. So around 10 nanometers, 100 nanometers आप जब कह रहे हैं तब आप complex की बात कर रहे हैं almost, you know. So around 10 nanometers, that's fine. So that means two molecules at that distance close by can have good Fred. Two fluorophores at those distances. But if you now increase the distance 10 times more, if you make it 100 nanometers, so वो 10 to the power 6 level पे आपका energy fall हो जाएगा. So 100 nanometers में Fred देखना, सारिका तुम एक बार cross check करो ये देख सकता है या नहीं. As far as my knowledge goes, it might be very difficult to see. But you have mentioned 10 to 100 nanometers when you were describing Fred. So
Ma'am,
क्रॉस
I
चेक एंड
used
टेल
it.
मी,
No,
क्रॉस
ma'am,
चेक को कहाँ पे ये दिखा कि 100 नैनोमीटर में फ्रेट होता है?
ma'am, I was thinking of angstrom in my mind, and I was speaking nanometer. So it.
ओके बट फिर भी एक बार क्रॉस चेक कर लेना, बिकॉज एज आई मेंशन ये exponentially fall होता है, and the decay rate is so high, it's to the power 6. Okay, all right. तो दूसरा है one was the electronic configuration that gives rise to the spectra. Second is the distance. Third, the term has something called resonance in it. अब ये resonance क्या है?
Ma'am, dipole interactions.
दैट इज हाउ सी, इससे ज्यादा रेजोनेंस मैं भी नहीं समझा सकती, क्योंकि इसमें जो फिजिक्स लगता है वो मुझे भी नहीं आता. सो जनरली हम लोग ये सोचते हैं कि एक का एमिशन हुआ, उस एमिशन को दूसरे मॉलिक्यूल ने कैप्चर किया, बट दिस फ्रेट इज अ radiation less process. There is actually no photons being released and captured. Instead, there is a kind of a resonance kind of a process where the energy is getting transferred from molecule 1 to molecule 2. So in reality, यहाँ पे जो ये fret हो रहा है, it's not because some photons are getting captured. Although हम वैसा सोचते हैं to make it simpler to understand. But you have to understand the name fret, the Foster distance आता है. Resonance, there is a resonance phenomena that's causing this, and there is energy transfer taking place. And the three important parameters are that you need to have a matching of the emission of one molecule with the excitation spectra of the other molecule. That is determined by its electronic configuration, and that also determines the resonance between these two molecules. They should be in close proximity. If they are not, then the energy transfer would rate, the energy transfer, the energy that can be transferred would fall drastically in the 10 in r to the power 6 order. So these are the things that are required for fret. So यहाँ पे हम इतना तो समझ सकते हैं कि फ्रेट इज अ वेरी गुड टेक्निक टू अंडरस्टैंड इफ टू fluorophores, two dyes, two fluorophores are in close proximity. If they are in close proximity, जितना वो close proximity में आएंगे उतना energy transfer बढ़ेगा, and then as they move apart energy transfer कम होता जाएगा. So if you can I saw a video where somebody was giving analogy of a butterfly. So you think of butterfly wings, and butterflies flap their wing, right? They go like this. So think of the tips having these two fluorophores. Every time they move close, you would have greater energy transfer because distance kam hota hai. Every time they move away, the energy transfer would decrease. So what happens when they move close and when they go apart? When they move close, let me explain it through this. When they move close, now first scenario is the wings are moving close. The distance between the two fluorophores is decreasing. जब ये होता है तब आपका जो emission है from one molecule, that would reduce from molecule 1, which is the donor. But the emission from molecule 2 would increase because now you have more energy transfer taking place. So instead of seeing these photons, you would end up seeing more of these photons. So as the distance between the two fluorophores keeps decreasing, you would see a gradual decrease of this emission and a gradual increase of this emission. Conversely, now if the butterfly is moving its wings in this direction, so distance between the two fluorophores is steadily increasing. If it is increasing, then you would have less energy transfer, which means you would keep increasing the emission photons from the molecule 1. You would see more photons being emitted, no energy transfer taking place, more photons being emitted from molecule 1. And because there is less of energy transfer taking place, you would have less emission from molecule 2. So till here, any confusion? I'm running out of time. Any confusion? Is it clear to everybody? Is it clear? ठीक है अभी
Yes,
तक?
ma'am.
ओके प्लीज रेजोनेंस समझने के लिए आपको किसी फिजिक्स के प्रोफेसर से ही ये सुनना पड़ेगा क्योंकि ये मेरे एबिलिटीज के बाहर चला जाता है. आई कैन गिव यू एन ओवरऑल व्यू बट आई सजेस्ट ऑल ऑफ यू टू टेक अप अ कोर्स इन बायोफिजिक्स. So here is the plan. Single molecule FRET. Very, very beautiful experiment. What did they do? They took a glass slide, glass nahi, quartz slide liya hai yahan, and they were able to, by some chemistry, attach this DNA, which is a stretch of 40 thymidine residues. So पिछली बार 16 थे, यहाँ 16 था, 16 को कम करके बढ़ा के उन्होंने 8, 6, 8, 10, 18 किया था. यहाँ पे उन्होंने straight 40 कर दिया. 40 stretches of this, and at the end 18 stretches को double stranded DNA बना दिया. So which means the rest of it, 40 minus 18 is single stranded, the rest 18 is in a double stranded duplex. Given this structure, the PCRA would bind at the junction of this double stranded and single stranded site. So exactly जहाँ पे double stranded खत्म होता है, single stranded शुरू होता है, उस fork पे जाके PCRA bind करेगा. So this is the reason why they have chosen such a design. You have a 40 nucleotide stretch and then an 18 nucleotide which is double stranded. You have PCRA that is bound at the junction where the single-stranded DNA is beginning because it binds to this junction. It likes to translocate on single-stranded DNA. The translocation is from a 3' to 5' direction. So ये जो single-stranded DNA इस तरफ है, इसका ये 5' end होना चाहिए क्योंकि it needs to translocate DNA and move towards this 5' end. तो यहाँ पे फ्लोरोफोर्स कहाँ डाल सकते हैं अगर हमें देखना है तो फ्लोरोफोर्स हम यहाँ तो डीएनए इस साइड में डीएनए मूव नहीं करेगा क्योंकि आपने यहाँ पे 5' एंड डाला हुआ है and it translocates on single stranded DNA. So it pulls single stranded DNA, translocates single stranded DNA. So single stranded DNA पे अगर ये मूव करता है तो हम यहाँ पे एक फ्लोरोफोर डाल सकते हैं और यहाँ पे एक फ्लोरोफोर डाल सकते हैं इस तरीके से कि ये दो फ्लोरोफोर्स known FRET pairs हों. If they are known FRET pairs, then what is it that we expect? We expect that, okay, this image will let you explain this. So what we expect is as the PCRA moves towards the 5' end, it could loop the DNA through it. हमने यहाँ previously जो ये image देखा था, यहाँ पे हमें दिखाया गया था कि ये straight away यहाँ से move करता जा रहा है. But this is because this DNA is kind of free, it's not attached anywhere. But if it is attached, the possibility is that it's actually going to loop the DNA through it, loop the single-stranded DNA, pull it through it. As it keeps pulling it like a rope, as it keeps pulling the rope, it will bring the end, the 5' end close to it. So this is a translocase activity, ATP-dependent translocase activity. As it keeps pulling the 5' end towards it, it is actually pulling this fluorophore towards this fluorophore. So as this reaction proceeds, you should see what? Ye butterfly ke analogy. You should see the two wings of the butterfly coming together. Two fluorophores are coming in close proximity. So FRET का सिग्नल बढ़ेगा. FRET का सिग्नल बढ़ने से क्या होगा? FRET जैसे जैसे आप देखते जाएंगे फ्लोरोफोर 1 का एमिशन कम होता जाएगा, फ्लोरोफोर 2 का एमिशन शुरू होगा और बढ़ता जाएगा. This is what you expect. So far, it's okay?
Yes, ma'am.
Yes, ma'am.
Alright, so now this same system has been taken, and if you do not add the PCR, if you do not add this protein, तो फिर क्या होना चाहिए? कोई ट्रांसलोकेशन ही नहीं होगा, ये दूर ही खड़ा रहेगा, the fluorophore. So you should be able to see distinct fluorescence from the two molecules and at the same level. ऐसा नहीं है कि एक का fluorescence कम हो रहा है, दूसरे का धीरे धीरे बढ़ रहा है, ऐसा कुछ नहीं है. दोनों का एक ही लेवल पे fluorescence है और FRET नहीं दिख रहा. फ्रेट का जो सिग्नल है, फ्रेट का सिग्नल मतलब ये वाला सिग्नल जो बढ़ रहा है, ये आपको नहीं दिखेगा. That is the condition if you do not add PCR, do not add helicase. If you add helicase now, you would have this fluorophore gradually being pulled towards the other fluorophore molecule because of the looping of the single-stranded DNA through the helicase, and that is represented by this increase in the acceptor fluorescence and a decrease in donor fluorescence. मतलब ये वाला green signal कम होता जाएगा, red signal बढ़ता जाएगा. You see the same thing, red signal बढ़ रहा है, green signal कम हो रहा है. And if you quantitate it in terms of the amount of red that you are looking at, that is the amount of this, you kind of make a ratio between the fluorescence emission of one fluorophore versus the other fluorophore, you can see that it keeps increasing, but then it falls. So why do I see this periodic oscillation? I would have expected कि ये बढ़ के एक जगह stop कर जाएगा, but ये नहीं हो रहा है, ये बढ़ के फिर से एकदम से fall कर रहा है. The reason is that PCRA, if you just take the protein out of the cell and try to look at its helicase activity, it does not have a high processivity. So कल सारिका ने processivity के बारे में बताया था, that processivity means how long can this molecule stay on the DNA, translocate DNA and cause opening up of the double-stranded DNA without falling off from the DNA. How long, how many nucleotides can it do this? PCRA का जो ये activity है, in vitro, जब आप अलग से सिर्फ PCRA को देखते हैं, ये बहुत low है. Inside the cell, when it is in combination with other proteins around it, then its processivity increases. So if you're just looking at PCRA molecule, इसका processivity कम है. इसी वजह से at the time it reaches the end of the 5' end, it simply falls off. When it falls off, there is nothing holding these two fluorophores at close proximity. Single-stranded DNA और भी ज़्यादा flexible होता है, and then the distance increases, and that's the reason you see a sudden drop. So you see an increase in FRET and then a sudden drop. Then the PCR reloads, again an increase in fluorescence, a sudden drop. So this kind of oscillatory behavior goes on in the system. Okay, तो यहाँ तक कोई question है? I just have one last slide to present. यहाँ तक कोई question है? Any questions so far?
Ma'am, what does this blue line represent in the graph?
The blue line presents what is called the FRET here, which is measured by looking at the fluorophore emission of the acceptor divided by fluorophore emission of the donor. So very simply, so FRET में क्या हो रहा है? आपका ये intensity कम हो रहा है ना? Because there is energy transfer happening, and the energy transfer is causing your molecule 2 to be excited, and the emission intensity goes up. So if you now take a ratio of this versus this, the more the energy transfer, the greater would be the ratio between this and this.
Yes, ma'am.
So this is what is represented.
Okay.
It's an easy way to represent that you are actually looking at a process of FRET. So you're not just giving the individual channel signal कि molecule 1 का emission देखो, molecule 2 का emission देखो molecule 1 का emission कम हो रहा है, molecule 2 का बढ़ रहा है, but you can also do a ratio and then that would show you very beautiful peaks. And यहाँ पे oscillation दिख रहा है क्योंकि 5' end पे जाके PCRA simply dislodges because it has very low processivity. It cannot stay very long. It dislodges, and when it dislodges, the distance between the two fluorophores increase. Again, the whole cycle starts.
Okay, ma'am.
So with this, they did a few of these calculations, and what it in the end it shows is that they have, so this experiment that they started with was with 40 nucleotides of DT. But then they also changed this nucleotide numbers. And the idea is that if you keep increasing the length of the single-stranded region in this very system, इसी system में if you keep increasing the length of this single-stranded stretch of DNA, then you would have more, what should I say, you would, how do I say, it would take sort of longer to reach the end of the 5 prime end for this as the DNA is looped. Let me go back here, otherwise I'm finding it difficult to explain. So this is say a 40 nucleotide stretch. If you now make this say 80 nucleotide, you double the length, then it should take double the time for this molecule to now reach in close proximity to this molecule. So therefore, your FRET curves should also show that it is taking double the time to reach the maximum FRET energy transfer. And if you now plot the average translocation time, how do you know the translocation time? Simply by looking at the time taken for this FRET to slowly reach the peak. You can understand what is the translocation time if you look at these in how do I explain this? Let me try to think about it. So if you take one of this curve, not the entire, सारे जो oscillating curves है सभी को मत लीजिए, आप एक ही curve को लीजिए. And now if you see how long it has, how many nucleotides it has moved in what time, and if you now plot that on a graph, you would see that that also gives you a straight line, similar to what was seen previously. यहाँ पे भी एक straight line था, that the more nucleotides you add, the more phosphate is released. And here you're saying the more nucleotides you add, the longer it is taking for this PCRA to reach the end of the 5' end. So this sort of gives you how many nucleotides your helicase is translocating per second from this graph. It's okay? Is it okay so far?
No, ma'am, I am unable to understand this.
Okay, so you have made an observation that if you have say 40 nucleotides, your FRET curves go like this. यहाँ इस distance पे आपको 3 curves दिख जाएंगे. But अगर आप इसको 80 बना देते हैं, तो maybe you'll just see for that to reach, you will see 1.5, which means it's taking twice as long. Now, if you make a plot between एक एक्सिस पे आप डाल दीजिए कि कितने न्यूक्लियोटाइड्स ट्रांसलोकेट हो रहे हैं और कितने टाइम में आप वो फ्रेट मैक्सिमम पीक आप देख रहे हैं, कितने टाइम में. So that is going to give you a straight line. So you can, from that straight line, you can sort of calculate कि per second आप कितने न्यूक्लियोटाइड ट्रांसलोकेट कर सकते हैं. Because you are looking at this FRET, your FRET data is being recorded in real time. So you know the change in FRET intensity with time. So वहाँ से आपको time के respect में intensity change का एक idea मिल जाएगा. And you also know how many nucleotides you've added because that's in your system. ये आपने design किया हुआ है. So how many nucleotides versus the time, and you would be able to tell per second आप कितने nucleotides translocate कर रहे हैं. It's almost like you're making a standard curve. Almost like making a standard curve. जैसे standard curve में आप different concentrations of DNA लेते हैं या different concentration of protein लेते हैं, and then you measure something. Here you're measuring the maximum say FRET energy that is given and the time it takes to reach that.
Okay, ma'am.
Yeah. Parman, you have a question?
Ma'am. Okay, ma'am.
Oh, you had asked
Am
the
I
question.
audible?
Ma'am, I just got confused. All right. So, from something similar, but I exactly did not understand how they came up with this calculation. I tried, but I could not. I'll try to see if I can check how they got to this. They also came up with another calculation that gives them the step size. That is uh, for for this helicase to move through this 40 nucleotides, how many steps is it taking? And in their calculation, it's all mathematical calculation where they have fitted an equation which is given here with the curves that they've observed here. Exactly how they got to it, even I'm not quite sure. What they found is for them, it was 33.6, so nearly 34 individual steps to be taken to finish 40 nucleotides. So it cannot be that 34 steps to complete 40 nucleotides. It's a rough approximation. जैसे मैंने कहा, it's not an exact measurement. It's actually an extrapolation and a calculation based on their data by fitting certain equations. So they have made some assumptions on that. So those are some limitations when they were doing this calculation in the first place. So 34 nucleotides, 34 steps to move 40 nucleotides को अगर आप realistically सोचते हैं, then it seems that it is taking maybe 40 steps for 40 nucleotides, which again comes back to the same thing that helicases, they take one step per nucleotide. At least this molecule is taking one step per nucleotide. The step size है इसका one nucleotide. And from the previous experiment where they had calculated zero point 0.5 ATP per nucleotide, so I said even that is kind of an approximation. So we understand that it is 1 ATP used per nucleotide. So 8 nucleotide translocation है उसका one step, and this one step requires the hydrolysis of at least 1 NTP. So these are approximate calculations. As you can understand, 34, 33.6 steps. ये एकदम direct calculations नहीं है. Maybe more direct calculations can be obtained by some other methods. But Fred could give them an approximate calculation कि कितना हो सकता है. So with that, I will stop today's class. And if you have any questions, anything that we have studied so far, anything on Fred, anything on these two papers, or you want to add anything extra about helicases, I'm here to listen. So yeah, it's 9, so maybe I'll stay around 2-3 more minutes if you have something to discuss. No? So I will request you all to take a course in biophysics. It's very important for your basic understanding of biology. Biology is very far from the biology that you have studied in your school. School level में आप जो पढ़ते हैं, ये kingdom है, वो phyla है, वहाँ से biology अभी कोसों दूर है. So biology now has entered a realm where if you are a biologist, you need to be good in computation, you need to be good in understanding mathematics and physics, even to some degree chemistry. So a basic course in biophysics is required for all students. Please do make an effort to take a course in biophysics. It will help you in understanding what you're doing in future. By the way, I don't run a course in biophysics, so ये मैं इसलिए नहीं कह रही हूँ कि मेरा खुद का कोर्स है, मेरा कोर्स नहीं है. आपको फिजिक्स डिपार्टमेंट में, किसी भी डिपार्टमेंट में अगर आपको बायोफिजिक्स का कोर्स दिखे, प्लीज कीजिए, even if you're auditing the course. Any questions? Anything that needs to be discussed?
No, thank you, ma'am.
Okay. All right then. If there are no questions, then we'll stop the class, and yeah, we'll see you again next Tuesday. Thank you.
Thank you,
Okay, ma'am.
ma'am.
Thank you.